I would first want to thank you for giving me the opportunity to share this work and hopefully get some feedback. I'm Ariel Erijman from New England Biolabs, and in this analysis, I compared germlines from different data sets in immune sequencing analysis. My goal is to show you New England Biolab Immune Seq workflows to give an overview of what we see as complexities in AIRR analysis, and I will compare between the germlines OGRDB and IMGT. New England Biolabs, I would define it as a community of scientists working to enable science in the world. We basically produce enzymes and kits so that scientists can focus on their research questions. And we have pride in our core values that are sustainability, transparency, and reproducibility in science. So our experimental workflow consists in two main steps. First, selecting the RNA from the samples. This is done with the bioethylated poly-T primers in a reverse transcription reaction, where UMI is also. Can you see my pointer?
Yes, we can see it.
I will use it this way. Where a UMI is integrated into the sequence through a template switching reaction. And then the B and T cell receptor sequences are selected using specific primers in PCR reactions. Then the rest is regular library preparation for next generation sequencing. So the reads look like this. In read 1, we see part of the C region onto the VDJ area. And in read 2, we have the UMI. Each UMI corresponds to a transcript. And we have generally enough overlap in between the two reads to generate the full sequence with enough confidence. In our website, we share a Galaxy workflow using Presto. We also have a Nextflow short script that can be used. And from Presto, we normally do the selection of filtering through quality, primary identification, removal of these sequences where we don't see those. We group through UMI, assemble the sequences, and we normally finish at the step where we have the final repertoire. We don't go normally into the biological questions. In this work, though, I used a Singularity container from the Immcantation portal. So while collaborating with the research groups, we had a few questions that we wanted to answer. The first is basic, like why do we assign genes and alleles to immunoglobulins? And the second one is what available germlines are out there, and what happens if we use one or the other? What difference do we see? Answering the first question was enough with asking and reading some literature. So knowing the theoretical and the observed combinatorial space, we have hints on how the selection process works in immune development. So we can track clonal expansion, we can associate alleles with disease, do evolutionary studies, and etc. We are convinced that this is a very important thing to do. However, it's complicated to map these hybrid sequences to the genome due to these regions being in short areas of the genome and being very, very similar to each other. As an example here, so this is what usually would happen, that we have ambiguous mapping, because in this case, the same V region happened to be almost the same. In fact, in this slide, I have the IGH, all IGH genes span about 1 million bases in chromosome 14. And in this heat map, I'm comparing all IGHV genes against each other, colored by their sequence similarity. And so you see that nearly all of them are at least 50% identical. I amplify this region, and you see that the values are really, really high. They are very similar to each other. The nomenclature, and I apologize if this is too obvious for many of you, but it goes from, we have the locus, the gene type, the cluster, and the position within the cluster. But also we have the allele information, and actually I would be very interested in knowing how often the allele information is essential to reach conclusions in these studies. We looked for available germlines for BCRs, and I focus on the first two. IMGT is the most mature, but it is maintained by only 2 institutes, and it has a license for commercial use. On the other hand, OGRDB is a collaboration from a worldwide community, the AIRR community, with plenty of institutes and labs, and it's open access. It looks a lot more collaborative, and we are interested in keep using OGRDB. Here we are testing it against IMGT. It also, I should comment that OGRDB uses VDJ-based information. So comparing naively just the number of genes and alleles in each of these germlines, these are the numbers, and we have a Venn diagram here comparing. So since if I split by heavy and light, I got about the same picture. So here I'm showing all genes and alleles from all regions, VD and J together. IMGT has a lot more genes and alleles, and therefore the intersection is not very big. However, when we exclude the allele information, we reach up to 66%, and also the IMGT genes and alleles not included in this case genes, sorry, not included in OGRDB, more than half are pseudogenes. They might almost hardly be expressed. We found it in IMGT, not in OGRDB, a lot of duplication. So in fact, all these listed genes and alleles from both columns are equal to each other, 100% identical. In fact, in the nomenclature, this D is indicating that is duplication in a distal area compared to the previous one. Sometimes even they belong to different clusters, but are exactly the same sequence. So we got PBMC samples. These are samples that have a lot of IG sequences, and we prepare libraries using NEB Immune Sequencing Kit. We sequence them in NextSeq 2000 and Element ABT instruments, and we reach about 380 million reads. Just to explain what I'm talking about what is a unique clone or counts of UMIs or transcripts. My apologies if this is too simple, but it has to be done. So here we have a sample with five transcripts, but we have only 3 unique clones, each defined in this case by the color. And this unique clone has 3 counts of UMIs. So with that, we analyzed the data from our samples using Presto, IGBlast, but and parsed with ChangeEO, but using either IMGT or OGRDB Germline databases. And we defined a clone as the combination of V and J regions and the CDR3 amino acid sequence. So these are the numbers of unique clones that we see in the samples. And by using either IMGT or OGRDB, we shared about 80% of this clone definition. When we reduced the information, excluding the alleles, we reached a shared group of 93%. So we dissected this into the V, J, and CDR3 regions separately. And so when we look into what we've used from these databases, in IMGT, we have more genes and alleles, and more V regions were assigned compared to OGRDB. In fact, the overlap was rather small. However, interestingly, when we look at the transcript counts or UMI counts, the correlation is really, really good. And in fact, for example, when we use OGRDB, so I must say that each dot is a V region, right? And the x-axis are counts of UMIs in log space. So when we use only OGRDB, for example, we had these 22 assigned V regions for which for IMGT we have no counts. And there is just a pseudo number added to avoid division by 0, but we got only 2% of these transcripts correspond to this non-overlapping group. And conversely, for IMGT was only 7%. So the overlap was actually very good if analyzed by the counts of transcripts. For J, we did the same. We have a lot less J regions, though, to analyze, and it's easier. And the overlap is rather good then. Fortunately, we have only 8 non-overlapping assigned J regions that we analyzed. And in fact, what we see is that these are, as we expected, though, these are just single mutations, in some cases maybe double. But so these are very, very similar genes that in case of IMGT, for example, for each of these 3 groups that I'm showing, in IMGT, we have both alleles in the database, whereas in OGRDB, we have only one of them. When we checked what's in common analyzing with IMGT or OGRDB for the CDR3 sequence, we would expect to have 100% identity, and we are almost there. The only difference is for cases where, for instance, there was a misassignment of a V region that presumably did not allow to find the cysteine or tryptophan and phenylalanine and J regions to generate the CDR3, and therefore we could not compare it to using the other database, but in general is extremely good, 99.6. So in conclusion, I briefly overviewed the workflow with our NEB Next kit. We validated using OGRDB in our analysis compared to IMGT, and the differences are not dramatic in our view. In this work, though, we use very highly diverse samples, and we might continue checking with less diverse samples to see which difference do we see. OGRDB currently includes only BCR germlines, but we will be very happy to validate the TCRs as long as they are present. Thank you very much. Thank you to Brad. He's the director of bioinformatics here. Chen is the developer of the immune sequencing kit with Evan Janssen. And thank you to Wenming Xiao from the FDA and all the people from the PCR project that we regularly discuss, and I learn a lot from that. And thank you to the NEB leadership for allowing me to present and discuss these topics. Thank you.
Thank you, Ariel. We have a question on the chat. Corey Watson is asking, when IMGT results in a call that you don't find in OGRDB, how confident are you that those assignments are actually of high quality?
Could you repeat the first part of the
Yes,
question?
sure. When IMGT results in a call, IMGT identifies a gene or something, but OGRDB doesn't. How confident are you that was correct?
For a gene, you mean a V or J region, or the definition of
maybe
clone?
Maybe Corey Watson, if he can unmute and clarify, or region. He said region on the chat.
Okay. So if let me try to share again. Sorry that I. So when in cases like this, we detect a V region that is not in the other database. The confidence, I'm not sure I can make an assertion on this. I can bring an idea. I don't know. I'm not. I don't know.
Yeah, but maybe, I mean, something interesting to follow up. It's an interesting
No, because the confidence, the problem of the confidence is that, so we have e-values for the confidence of the assignments, but I'm not sure. I did try to compare. I don't know if he meant he was referring to that. So I did try to do these comparisons, but these e-values were a little bit noisy at some point. So they go either to the extreme or they remain close to zero when there's not good confidence for the assignment. I don't know, was that the question, or I didn't understand?
Yes and no. I basically, like IMG, I think the point here is to think about is IMGT is a bigger database, right? And sometimes when you have more sequences in your germline database, you will just get more calls. But that doesn't mean that those calls are legitimate. It just means that if those are, for example, poor alignments, then you may be getting that call just because your sample is lacking a true allele. Excuse me, your database is lacking a true allele that your sample has.
So the problem with that is that even if I compare to the sequence group by UMI, for example, where in the consensus I know very well if a mutation is actually what I should be seeing, I still don't know if should be that the allele present. Maybe in the sample we do have the other allele. I'm not seeing it in this transcript. Am I clear with what I'm saying? So I don't know. I don't have that confidence.
I mean, yes and no. I think it's something to watch out for, I guess, is maybe what I would say. Right?
Yeah, I
Because
would be happy to discuss this.
when you have a database that's bigger, it doesn't necessarily mean it's reflecting reality, right? So
Right.
you're basing assignment means this is the best match I have among the sequences, right?
Correct.
So I think it would be like, are all of those, say, are you hitting those assignments with 100% identity to that germline, for example? So I think there might be some stuff to look under the hood here. I mean, this is something that we're all doing. But I think it's an important, when you're conducting a comparison like this, I think that's an important thing to consider, right? Because assignment is a sort of, in many ways, it's a loaded term. It's just like the thing that the aligner told you. But that
Right.
could mean many things.
Yeah, the problem is that single mutation does not vary the only value that I can use for confidence, which is the value, enough to claim what's right and not. But I would be happy to ask you later on about this.
Sure, yeah, happy to chat.
Yeah, I think there's a big question for the community, the issue of germline databases, and definitely we should keep discussing here and at the air community meetings and other forums. So really, thanks for a great talk.